Yeah, so our next speaker is Yaqun, and she will talk about the role of Topoisomerase I. And I cannot see my notes anymore. Attends. C'est un peu relou quand... Ah. Voilà, the role of Topoisomerase I in preventing transcription replication conflicts. So Yaqun, you should have the microphone now.
Yes, hello. So can you see my slides now? Sorry.
Yes, it's fine.
Okay, so thank you for the presentation. Okay, so hello everyone, my name is Yaqun Liu. I'm a second year PhD student from Team Chen, which is Replication Program and Genome Instability of Institute Curie and PSL Research University. So today I'm very happy. Thanks a lot for the JOBIM organization committee to give me this opportunity to let me present our recent work about the Okazaki HMM, which is a genome-wide folk directionality analysis toolkit. And I'm gonna also present its application on transcription replication conflict study. So I will start from some basic biological background about the DNA replication and Okazaki fragments. So the entire DNA replication process is considered semi-discontinuous. So to zoom in a replication fork, it contains a leading strand, which is replicated continuously, and a lagging strand, which produces the Okazaki fragments in a discontinuous way. So Okazaki fragments are short sequences of DNA nucleotides, around 150 to 200 base pairs in eukaryotes, synthesized discontinuously and later linked together by the enzyme DNA ligase to create the lagging strand during the DNA replication. Since the Okazaki fragments only show on the lagging strand, so we can use it to measure the replication fork direction. So in collaboration with Olivier Yehian's team at ENS Paris, so a new protocol to sequence the Okazaki fragments of human cells is successfully established. Basically, the nascent DNA sequences are labeled by EDU, then small DNA fragments less than 200 nucleotides enriched with Okazaki fragments are selected. And finally, the biotinylated fragments were captured with streptolidine, ligate the adapters, and then perform the single-strand DNA sequencing, which can keep its original orientation. So here is the replication fork directionality profile of HeLa cells. So firstly, we just get the read coverage for each strand. And then by calculating with this formula, we can get the replication fork direction called RFD. So the blue signals indicates leftward replicating forks, and the red signals present the rightward replicating forks. So here is a 1 kb bin size and positive slope of RFD profile just give the position of a replication initiation zone, since it presents divergent fork movement on its two sides. And a negative slope presents a replication termination zone, since the direction of two sides are convergent. Since the OKseq is now well developed, and more researchers just use it to study the DNA replication. In order to facilitate the data analysis, we decided to construct a bioinformatics toolkit for that, and we just named it Okazaki HMM, which is now available on our GitHub team. And so here it's the pipeline about how to use the package to proceed the Okazaki data. So firstly, the raw data is pre-proceeded to get the mapping and they duplicated data. So then use our OKSeq HMM, the data will automatically split it into forward and reverse strands, then calculated the read depths in 1 KB adjacent bin size, and then the 15 smoothing bin size. And finally proceed the replication initiation and also the termination zone calling by HMM. Second function is to calculate the delta of D to get the origin efficiency metric in multiple scales to see the, like we can say, dynamic RFD transition. And also the last function is to plot the average meta profiles and the corresponding heat maps for regions of interest. So the outputs such as RFD, OEM, IZs, etc., are easily visualized in genomic visualization browsers like IGV and could be used for further downstream analysis. So in our tool, we use the hidden Markov model of HMM to predict the replication initiation and termination zones. So you can see in a typical RFD profile, like origins cannot be directly observed like the hidden states. But the signals vary from -1 to +1 continuously with trackable patterns, which like the rightward patterns depend only on their neighbors' leftward patterns. So that's highly correlated with the HMM model application condition. And so we can extract this pattern to set up 4 states like the IZ and the up flat and down flat, the 2 intermediate replication processes, and also the TZ, the TZ, like the termination zone. So for the observations, we divided the RFD into 5 quantiles. By using a training subsets, we just set up the different transition probabilities among the 4 states, and also the emission probabilities between states and the observations to successfully, you can see here, get most of the IZs, the replication initiation zones, and also the TZs, the termination zones, in 1 KB resolution in human cells. We also compute the origin efficiency metric, the OEM, to visualize the RFD transition. So we like calculate the delta of D between the 2 extremities in a defined scale, like 10 KB or even larger bin size. And basically maximum of the OEM represents origins and minimum refers to the terminations. And also the degree of OEM is proportional to origin firing efficiency. Like here, like the different amplitude of OEM just represent different efficiency level of origins. Then we just go on for case study, like its application on OKSeq of different human cells. I collected almost all of the published OKSeq data of different cell lines and just reproceeded them with our OKSeq HMM, and all proceeded results are also available on our GitHub. Here is an example of RFD profiles of different human cells and generated by our toolkits, which is quite obvious that they are quite cell type specific. And then for the initiation and termination zone calling step. So this figure just show us the RFD and also the corresponding initiation zone, the IZs, detected by the HMM, and also the heat maps of OEM from 10 KB to 1 megabase window size. And we can clearly observe that the positive OEM in you can see that in red box. So just match to the IZs, while the negative OEMs in blue box are related to the TZs, which allow us directly visualize the IZs and TZs from the IGV. And for an average metagene profiles to see the OEM, so we can generate this kind of profiles around all of the detected IZs. Like we can see the human IZs normally range from 10 KB, then even to 100 KB length, with majority around 20 to 30 KB. And as expected, we can see that IZs show the strongest positive OEM at corresponding scales. And we can also plot the average average RFD profiles for all the reference and active genes. We can see that IZs enrich at like the transcription start sites, and also the TZs are mostly at the termination, the transcription termination at the TTS. So we can see at TSS genes are prone to like more co-directional between replication and transcription direction, while at TTS, like genes are prone to have head-on collisions. Yes, and we generate also the heat maps for RFD and OEMs, for example, around TSS, the transcription start sites. Like I just sorted by the gene size from short genes to long genes, as shown here. And we can observe that starting from TSS, we get the strongest RFD and the OEM signals. Then the RFD regularly decreases along the genes. Also, we can see a correlated decrease of OEM signals from 10 KB to 500 KB scales in the gene size dependent manner. So finally, I'd like to talk about how I apply our tool to solve a particular biological question, like transcription replication conflicts. So as we all know, like replication and transcription machineries share the same DNA templates, which can lead to collisions. So these collisions can occur either in a head-on or co-directional manner, but head-on collisions have been shown to be more deleterious. It might induce the genome instability via a special structure called R-loop, which is a DNA RNA hybrid. And so this is a metagene profile with R-loop signals in red, performed by the DRIP-seq, and also the Fock stalling signals in black. So we just get the Fock stalling positions by perform the phosphor RPA DRIP-seq. So we observe that R loops are formed both at TSS and TTS, while the replication Fock stalling signals are enriched only at TTS. And as you might remember, so by our Okazaki data, we found that at TTS, genes are prone to have more head-on collisions, which propose that the head-on collisions could induce more replication Fock stalling. So here we can directly check the corresponding data in IGV. So you can see for the gene MED15, the R-loop signals DRIP-seq are enriched both like at both sides, and the Fock stalling signals, the phosphor RPA, are only at TTS, where the genes are prone to have head-on collisions revealed by our Okazaki and RFD. Then we more focus on TTS to identify the, we can say, toxic R loops. So here are the two heat maps around TTS for control, HeLa cells again, and also their corresponding top one deficient cells. In fact, the top one is an important factor to help to resolve the R loop, induce the genome instability. So the genes are clustered into 5 subsets by different transcription level. Then we just check their corresponding R loop, foxeling, and also the DNA damage level, like obtained by the gamma H2AX tube-seq. So we can see that the top one depletion leads to DNA damage associated with higher level of R loops and foxeling of highly transcribed genes at TTS. And then we use the higher resolutive double-strand breaks mapping data obtained by BLESS technology. And I then separated the genes by their double-strand break level into two groups. So the highest 25% double-strand breaks level, and also the rest genes. So the profiles also indicated that the top one depletion favors the double-strand break formation at R loop enriched TTS regions. So here's the conclusion, and we finally constructed the first whole genome model to show the link between R loop and TRC, thanks to our tools. And here is all the conclusions I already mentioned previously. And if you are interested, you can check our papers to get more details. And for more sources available, in fact, our team members also develop different softwares like optical replication mapping, replication timing, common fragile site mapping, and also for the single cell, even for the single cell analysis tools, and all you can find from our lab GitHub for more details. And finally, I would like to thank all my team members and especially my supervisor, Chuanlong Chen, for all the help and support. I also would like to thank the platform for the technical support and also our collaborator, especially Dr. Philippe Bassero's team in IGH in Montpellier for performing all of the experimental data. And finally, I would like to thank all the fundings for supporting my PhD project. So thanks for your attention. I'm ready for questions. Thank you.
Thanks. Thanks for the talk, Yaqun. I just noticed we cannot upvote the previous clap. So somebody can make a new clap and we can all upvote it if we want to. And meanwhile, merci. Meanwhile, I will check. So we have time for a few questions. We have like 2-3 minutes.
So should I stop sharing or it's okay for now?
It's fine. You can. I think you can keep sharing. So if you have questions that need slides to answer, you have them.
Sure. Yeah.
So questions. Oh, Yaqun, I have one naive question just to get people started and make them not afraid to ask stupid questions.
Sure.
So I think you explained that TSS, transcription and replication was correlated, right? You showed correlation between TSS, transcription start sites, and replication sites.
Yes.
Yes, but then you also showed that replication sites seem to be tissue specific, right? So is it is a correlation only with the genes that are expressed in that specific cell type or something, or what?
Sorry, I just show here. Maybe I mean like here I just show that all the profiles are kind of cell type specific. yes. Right, but for the like TSS for the transcription start site or transcription termination sites, I think it's kind of conservative. It's always like we can see replication origins kind of enrich or more correlate to the transcription start sites. We can say here that's and termination, the replication terminates also more or like or prone to stop at the termination, the transcription termination site. Yeah, that's we find from our results. Right.
Yeah, but the question was, are the genes like particularly expressed in the cell types where the TSSs correlate with the IZs?
You mean the cell
in the cell types?
Cell type? No, I don't think they are cell types related. Yes.
Okay. So anybody else want to ask a question? If not, we thank again Yaqun, and we'll move to our next speaker.